Wonderful. So hello, everyone. I am Kimberly Housekeeping, the CEO at Global Genes, and I am so excited to welcome you all to the 5th Annual Rare Drug Development Symposium we hold in partnership with the Orphan Disease Center at the University of Pennsylvania. Wherever in the world you are, I just want to say thank you so much for joining us today. As a rare mom myself, I am so grateful that we are able to have this meeting and connect together and learn together today and tomorrow. We all recognize the incredible challenges we have had this year, without a doubt. And I absolutely speak for all of us at Global Genes when I say we continue to be utterly amazed by your dedication and drive to ensure that care and research for the rare disease community continues during these absolutely unprecedented times. Our resilience is strong. In rare disease, we deeply understand the importance of being able to connect with others. And as the COVID-19 pandemic spread worldwide, we listened to you and we made adjustments to ensure that we could stay connected and also protect the health and safety of our community. That's why we're in virtual mode today, and we're glad you're here. Our hearts are with you in person, though, please know that. But more than just making connections, our community is really built on a shared social pact. We stand together, we listen to each other, and we share our stories to build understanding and create hope. Hope for everyone impacted by rare disease. But there are still members of the rare disease community that are not seen, not heard, and not included, namely communities of color. We recognize that inclusivity and embracing diversity are absolutely critical to shortening the diagnostic odyssey, critical to advancing the disease understanding and translational research, and critical to developing more meaningful clinical trial data, and absolutely critical to strengthening the care and support for rare disease patients. It is our privilege and our responsibility to use our voice to advocate and break down barriers to a more inclusive and equitable world for the rare disease community. While this virtual symposium looks a little different, we are still so thrilled to be with you today. So excited. Each of us are here because of the rapidly evolving role rare disease patients and advocates have, where patients are partners and active drivers in research and therapy development. And we want you to leave here today and tomorrow with insights, with new ideas, tools, and with connections to advance your work. New to the symposium this year, I want to share rare rounds. These are roundtable discussions on key topics, and really key topics that are needed for the building and strengthening of rare disease research. I absolutely encourage everyone to join in these very important conversations over the next 2 days. And for our young researchers in the audience, we have added a rare research investigators workshop. And this will provide resources for researchers beginning or those that are growing in their rare disease career. I would also truly like to thank our many sponsors. Your continued support of this event is so incredibly, incredibly important. Thank you so much for believing in us, and without your generous support, we would not be able to provide such an extensive program today and tomorrow. We are grateful and glad that you are with us today. I also absolutely need to stop and thank our Global Genes team and the Orphan Disease Center for all of the hard work in putting this event together. Lastly, without a doubt, I need to thank a very, very special thank you to all of our speakers. Without your willingness to share your expertise and your willingness to share your time in the next 2 days, we wouldn't be able to have this amazing event. Now, as you explore the event platform and attend sessions over the next few days, please be sure to have a little bit of fun too. Keep your eyes out for the Global Genes ribbon. For every ribbon you find, you will receive points. The 3 participants with the most points at the end of the event will win prizes, and we'll notify you via email, and I wish you luck in this process. Have some fun. Now let's get things started. I'd like to welcome and introduce both Dr. James Wilson, the Director of the Orphan Disease Center, as well as Susan Brizendine, our Vice President of Engagement Programs at Global Genes. You may also know Susan as Susan Sikora. Susan has 25 years of experience leading international and national engagement and education initiatives. In her most recent role at the ARM Foundation for Cell and Gene Medicine, she was chief architect of the Global Healing Genes educational program. Personally, Susan is on the undiagnosed journey with her 10-year-old son and is absolutely passionate about equipping families with resources and encouragement to advocate for their health needs. Turning back, importantly, to Dr. Wilson. Dr. Wilson is a professor at the Perelman School of Medicine at the University of Pennsylvania, where he has led efforts to develop the field of gene therapy. His research career spans more than 40 incredible years and has focused on rare disease and ways to treat them using gene therapy. The Wilson lab identified a new type of vector based on novel isolates of adeno-associated viruses, and they have become the best in class for gene therapy and are used by 30 or more companies. It is truly amazing work. I would like to now turn this over to Jim to formally kick off our annual Rare Drug Development Symposium. Welcome, Jim. Thank you for being here, and let's have an amazing meeting. Well, thank you.
Oh, Jim, you're still on mute.
Good morning to everyone. And on behalf of my colleagues at the Orphan Disease Center, we welcome you to the symposium and delighted, Kimberly, that we're able to serve this community through our partnership with Global Genes. You had an incredible new dimension to what we were trying to do before. Indeed, 2020 has been a challenging year on multiple fronts. It's a year I don't think any of us will forget, including rare disease stakeholders. Due to shelter-in-place mandates, regrettably, many of the labs who were working on rare diseases were either shut down or activities substantially curtailed. Clinical trial, a key activity of what we try to do, were either reduced or stopped, and this has been frustrating for all. But I'm going to suggest today is the day of a new beginning for us, and and I think we should mark this symposium as that new day. Labs are reopening, as you know. We're no longer using the word essential research, which I think every research is essential, otherwise we would not have been doing it. And scientists are hungry. There's nothing worse than telling a scientist you can't do science. While we have not been at the bench, some of us have, many of us, I think all of us, have taken the time to reflect and to strategize, which is an opportunity we usually don't have in our frenetic lives. Hospitals are now accommodating non-COVID patients. Clinical trials are being rebooted. And the revolution in diagnostic and therapeutic technology platforms that has just created a new day for rare diseases are not going away. They've actually been maturing and ready to be deployed. Stem cells, RNAi, gene therapy, genome editing. And finally, which is, I think, one of the biggest surprises for me, investment in biotech companies and investment in research by the biopharmaceutical companies is at an all-time high, despite all of the challenges we've had in our economy. Share prices now are exceeding that we saw in the bull market at the end of 2019. So it is a new beginning, but I'd like to share with you some thoughts, personal thoughts about what I'd like you to think about over the next 2 days. I could probably use an hour to do this, but I'll keep it short. First of all, we need you. We need families, we need foundations committed to individual rare diseases. And you need to take the lead in moving the research bar forward, as I see increasingly and as I hope would come out of this meeting. The fact is that business models for developing rare disease treatments aren't particularly strong, especially if the chance of success is low and the number of patients are not many. As communities, you need to be smart in what you invest in and establish the foundation of science that allows companies to proceed with investments with less risk. And this will happen and will continue to happen. Despite these efforts, there will be diseases that are simply too rare to achieve the kind of engagement with drug developers that we need. We need to develop new models and consider new models. And I'm delighted to say that today and announce that a collection of stakeholders in the business and the scientific community have created a new 501c3 called the Institute for Life-Changing Medicines, whose goal is to develop and distribute potentially curative therapies for ultra-orphan indications. Stay tuned. Second, we need to direct our efforts towards the solutions that apply broadly across all patients living with rare diseases. It is my personal opinion as a scientist, been involved in this for as long as Kimberly, you noted, that is as easy to develop a more general solution to a treatment than a therapy targeted to a single individual. And then finally, when we get there, and we are, let's make a commitment to the entire rare community to strive to achieve global access. Genetic disease is not found in any one particular part of this planet. So thank you all for being here over the next few days, virtually, I'll say, and for your passion and commitment. We as scientists really feed off this energy. Enjoy the meeting and be safe, and Godspeed. Thank you.
Thank you, Jim. And thank you, Kimberly. And thank you for the partnership between Global Genes and the Orphan Disease Center. So many challenges in our world right now, as you've said, and so many opportunities too, hopefully for lasting change. Hello, everyone. My name is Susan Brizendine. As Kimberly said, I lead engagement programs at Global Genes, and I'm thrilled to be part of this welcoming committee this morning. Pictured here are some pre-COVID photos taken during Rare Disease Week in Washington, D.C. Who knew that that week would be the last chance to really hug each other for a very long time? And now we spend most of our time in these little squares wishing we could just reach out and touch someone. But the smiles and the hope, they keep us going, right? As Kimberly said, the rare disease community.
Susan,
Yes.
I just wanted to say, it might be easier for everybody if you switch your screen presentation. Right now, there's a lot up there because everybody can see the notes as well. So might be.
So sorry. Okay. So let me just figure out how to do that. I'm sorry. Does that work?
That looks better.
That looks there.
Okay.
Thank you.
Okay. All right. So I just have to switch gears for just a second here. So what I was saying was that the rare disease community models resiliency, and we're humbled to walk alongside you and to serve you and to support you. I'm going to have to ask you, Maureen, to advance my slides, I think.
Sorry, Susan, I can't access your slides right now.
Okay. Okay.
I'm sorry.
Oh, goodness. Okay. Give me 1 second. Okay. So as Kimberly stated, you know, we responded to COVID-19 as many organizations did. And what we did learn is that, you know, the key things that emerged obviously were continuity of care and continuity of research. And the COVID pandemic, as both Kimberly and Jim said, it significantly impacted how care and research will be defined going forward. So, you know, what did we learn? How are we responding? And what does it mean for our time together over the next couple of days? Well, there's no question it caused a crisis, as Jim said. Access to care and access to research was restricted or denied. And the priority became, how do we very quickly elevate awareness of the unintended consequences for the vulnerable? So it became very urgent. And we assembled a multi-stakeholder task force. And based upon the themes that emerged around continuity of care and continuity of research, we, together with our partners, issued a call to action and urged a rapid resolution. So some of those issues that were raised in the call to action were the importance of focusing on clinical and psychosocial needs, so supporting data collection for partners and agencies, infusion services, transfusions, in-home nursing and support, safe treatment pathways in clinic, critical care access for high-risk patients. We also needed to focus on the urgent economic need. So we raised funds to support an extension of our rare patient impact grants to focus on continuity of care. And that provided urgent financial aid to rare disease organizations and families. And by the way, the grant opportunity is still open, so we encourage foundations to apply. We also looked at ensuring the recovery of research. So together with our partners, assessing the impact of COVID-19 on late stage and human cell line research, fostering direct investments in research and development with innovative new nonprofit models, supporting FDA-approved pivots to decentralization. And another area that was particularly important for us is educating advocacy organizations on what to do next. We convened foundation leaders to just brainstorm workarounds when, for example, they lost access to their research lab or their assets, shared encouraging perspectives from experts. And in talking to foundations, we learned what was really the key impacts for them. And they said, beyond some of the obvious that we've already talked about, it was the cancellation of events, the emotional toll of the anxiety and isolation brought on by some of the shelter-in-place orders and lost funding. So the resources they wanted from us were access to funding resources, and they wanted to keep their communities engaged in this remote world, and they wanted emotional support. So we created one of the first resource directories with help from organizations around the world that's been widely distributed, and we're launching a new topical community development leadership series called Virtual Rare Leader Meetups that are really designed to address the needs that the foundations expressed, and it's an opportunity for them to continue in some remote learning while enjoying some informal discussion and connection. But amid COVID-19 and beyond, our focus continues to be shortening the diagnostic odyssey for families, increasing the number of diseases that have research in the pipeline, and improving, ultimately, patient outcomes. And how do we do that? Well, first we need to understand challenges and barriers on access. We need to strengthen care coordination. We need to equip families and foundations with tools and resources. And we need to build a data-centric community and organizations. We have to do that hard work. And we do this by partnering with you, right? We're partnering with industry and foundations and with researchers. It's why we're here today. We're all collaborating to make an impact. So we have 3 alliances at Global Genes. We have a corporate alliance, we have a foundation alliance, and we have a research alliance. And they allow us to move nimbly in the fluid circumstances nature of rare disease research and development while aggregating data and sentiment to help prepare communities to really embrace the challenges of ensuring that treatments are approved and accessible. So the Corporate Alliance has more than 110 members now, the Foundation Alliance more than 600, and the Research Alliance is just forming their charter membership to begin addressing the gaps in life sciences. And we're here today with the Orphan Disease Center to inspire careers in disease research. Advocates, you're here to learn about the research options, the developments in the science that will lead to greater understanding of your diseases and how to foster collaborations that will propel your work forward. To our young investigators, you're here to pursue partnerships and to learn how to maximize your relationships with advocacy organizations to design and implement the most patient-centric research. In industry, as sponsors and arbiters of funded research, you have a crucial role in funding research that's inclusive, fosters diverse and robust data, and ensures better outcomes. Because at the end of the day, we will all stand together in front of regulators who will demand robust, reliable data rooted in sound, fruitful collaborations. So looking at our agenda, there are crucial updates from the realms of artificial intelligence and drug repurposing that are not only on the cutting edge of rare disease research, but are actually essential tools to the development of a vaccine for COVID, which is a rare infectious disease in its own right. So we'll be covering the importance of developing robust collaborations from both perspectives of advocates and researchers, and because we have to speak a common language and understand the challenges of both sides. And as you participate in the symposium over the next 2 days, we hope you'll challenge yourselves to think about what types of connections and alliances do I need to foster in order to strengthen my research efforts? How can I be a better research partner? And as you're sitting at a round table around diversity and inclusion, ask yourself, what do I have to do to improve our organization's inclusive strategy. So let's be challenged, as Jim said, to consider the global investment, the worldwide collaboration and urgency with which researchers are investigating COVID-19, and to think about how do we bring this same purpose and openness to our own work. We hope that you'll leave this conference feeling inspired to explore your role in rare disease drug development and in new and innovative ways. And we hope you'll feel empowered and equipped to continue to push forward on necessary treatments and cures despite the setbacks caused by COVID-19. And we look forward to continuing this conversation at Global Genes Live, a rare patient advocacy unsummit. You don't want to miss this event. Join us online September 14th through 25th. You can sign up to receive more information. Please connect with us. Visit globalgenes.org for more resources that I could possibly list and follow and tag us on social media so we can share your news. With all of that collaborative framework in mind, we're excited to dive into all the wonderful sessions and discussions and networking opportunities we have during this event. And before we get underway, I just have to do a few brief housekeeping notes. So for advocates, we have parallel sessions divided among a focus on foundational and expansion questions. So the foundational sessions are designated as 1.1, 2.1, and 3.1. And on the agenda, and the expansion sessions are designated as 1.2, 2.2, and 3.2. Those are the expansion sessions. And again, we have workshop sessions targeted toward academic researchers and how they can grow their rare disease efforts. Finally, for both advocates and researchers, we have exciting plenary sessions, research posters, and rare rounds, our roundtable discussions on key topics needed for building and strengthening rare disease research. So please refer to the agenda posted in the auditorium and in the lobby to select whatever sessions appeal most to you. So this afternoon, we'll be hosting Cures Within Reach and their 2020 Cure Accelerator Live for Rare Disease event. Cure Accelerator Live Rare Disease is a philanthropic pitch competition from Cures Within Reach that gives attendees the chance to vote for the next breakthrough repurposing treatment. Cures Within Reach's 5 finalists are principal investigators presenting their clinical repurposing projects for patients with rare diseases. If you have any questions about the agenda or the logistics of this virtual event, please see the help desk, and they can also give you some tech help there as well. So during the sessions, all attendees will be muted by the moderator to reduce any interruptions. We encourage you to use the chat function in the Zoom window for any questions you may have throughout the session, and then the moderators will pose those questions to the speakers. And if you, again, have any technical difficulties or questions about the program as a whole, please visit the help desk in the main lobby, where you can chat with our tech team and our Global Genes staff members. So now, the very best part, I have the privilege of—
Hey, Susan.
Yes.
Interestingly, we're actually running a tiny bit ahead of time.
Okay.
But we're just a few minutes ahead. And so Jim and Susan and Kimberly, if you wouldn't mind, I think it'd be great actually to open just to a few questions and a little bit of discussion before we get started with our keynote speaker, if that would be all right.
Sure.
And so please, anybody from the attendees, if you have any questions, I'm watching those and I'll be able to forward them on to the panelists. But in the meantime, I thought I would kick it off just by asking Jim to start. You know, what are some of the things that right now are really exciting for you in looking at rare disease research and some of the promising opportunities that are coming over the next few years? I'm sorry, Jim, you're still on mute.
Been schooled in the unmute world. Is various technology platforms are really coming of age, including gene therapy, which after 3.5 decades is there. And we can leverage the experience of the successes of Zolgensma, Luxturna, and many other diseases. It's time to put the pedal to the metal. And then genome editing will be another wave. But But I think one of the challenges is going to be, as we bring these modalities forward, in ways in which we can conduct clinical trials without control arms, which you can't, is natural history registry data. This is a huge, huge gap that we have to figure out. In each of your communities, it should be on the top of your agenda when you come together to acquire those data, so that when companies or scientists come forward with interventional opportunities, that they have the data to compare against which they're going to study whatever their treatment is.
That's a really wonderful point, Jim. And would just to expand on that just a little bit, this is much more in depth than registry, contact information that you're talking about for the natural history study that could be used in order to potentially provide some placebos or just some understanding of the disease, correct?
Exactly. You've got to acquire clinical data relevant to the progression of the disease, which can be done as a protocol, formal protocol, forward-thinking, prospectively acquired data. That's not practical in most diseases. We've got to identify ways to extract from medical information that's acquired as part of standard of care enough information and assemble those data in a way in which we don't have to set up and stand up a completely new prospective study. There's a lot of data out there about individuals living with rare diseases. We have to find a way to acquire it, and globally as well.
And I apologize to everybody. I just wanted to take a moment. I realized I haven't introduced myself. I'm Morgan Hart. I am the Director of Research at Global Genes, and welcome everybody. Really excited about this event. This is one of my favorite events all year with the partnership with the Orphan Disease Center. So Jim, very quickly, one of the questions came in that is really quite interesting, actually, and I'd love to get your take on that too, around what kinds of new regulations might be needed, if any, or can we work within the current regulatory framework and just adapt it in order to really accelerate rare disease research?
You mean these are health authority regulatory reform? Quite frankly, we've had pretty good success working with regulators, FDA and ex-US, and quite frankly, I think we have to be careful not to push them too hard, because we don't want to bring products into the market that aren't proven to be safe and effective. But I tell you, the most compelling argument to the regulators are your stories, and that has the largest impact in terms of understanding how best to proceed. So whatever mechanism you can through your communities to reach out to health authorities so they get to know you, and there are formal mechanisms and informal mechanisms. That's just priceless, and it's something that only you can do, the scientists and the companies can.
Great. We actually have another question too. One of the things that has really developed and expanded within our current climate with the need to adjust with COVID-19 restrictions. And so one question from the attendees is, what do you think FDA might be open to with respect to telemedicine approaches for natural history studies?
Oh, I think it's a great question, and we're already pivoting to that as well. I mean, there's certain tests or examinations, imaging, for example, that you can't necessarily acquire through telemedicine, but especially globally. I mean, in order to achieve data from different communities throughout the globe, it's probably the only way that it's going to happen. But COVID has accelerated a process that was probably already underway, and I think in the other end, we're going to benefit from that in a way to acquire those data.
Great. We do have one other question about which registries you use to assist in the process. And I think instead of that, Jim, if you might be able to expand, and I know every disease is a bit different, so there's a challenge in thinking about trying to dictate one thing for all rare diseases. But what kinds of data are you talking about within these natural history studies, just very broadly?
Yeah. Well, you know, at the highest of level is what we call a patient portal, where we invite patients and their families to participate by entering in basic information about demographics and the kind of information that a family can provide. That's a great way to start, because essentially you know who is out there, and this is a vehicle to communicate with them about new trials. The second is the Research Registry, which I think is really where a lot of the business should be, and where it's affordable, to find a way to work with the healthcare providers and the patients to acquire the data that exists in patient records in hospitals and in clinics. And it's been acquired in a way that addresses specific research questions. How best to do that? A lot of different models. With the electronic medical record, it's become a little easier to do it. But I think there's opportunity for creativity in that space.
Wonderful. We have a few more questions from the audience, just really quickly, the attendees. So we're talking an awful lot about data. That's really going to be so terribly critical, Jim, as you're mentioning, within as we move forward. And there will be, you know, we're really working to build collaborations within the Orphan Disease Center. We see that Global Genes, of course, is helping to identify new paradigms for collaborations that are going to include all stakeholders all at once, industry and patients and academic researchers. Jim, do you have any thoughts on how we can balance the needs of the stakeholders and be able to provide this data as broadly as possible?
It's a really important question, because we live in the reality of those communities which are competitive. Competition actually drives progress, and we have to know where to define what's competitive and what's not. I think working together to determine what's pre-competitive space. How can we work together to bring information together, data together, for example, registry information, certain standards in which we measure things. And then if we can define that we can all work together to benefit all of us. On the other side of that line, we've got to let companies proceed with their business and develop products in which they're going to be competitive with one another. I think there's a limitation to what you can do there, but the world of what is pre-competitive space is growing and growing as these communities are maturing.
Absolutely, I think that was very, very well said. And following up on that, and again, kind of thinking about how COVID and the pandemic has really changed some of the mindset across all of really biomedical research and care. There's been so much collaboration around COVID, and Jimmy mentioned a lot of the labs were pivoting to kind of focus on that. And so we have a question, you know, with all of the efforts and the collaborative efforts that we've seen as they really, as researchers have really come in together within COVID efforts, what kind of lessons might we take from this for rare disease? Are there, you know, some thoughts around how we might be able to leverage this precedence for rare disease?
I think the way in which we share and communicate with one another. I gave a talk at a symposium for an educational session that the room would have held 20 people. There were 1,500 people listening to my talk. The kind of reach and impact that you can have is just enormous now. And maybe this would be an example going forward. With that said, I miss you guys not being together. I miss at the end of the session going out for coffee and bumping into people. And I think there's going to be a balance between you know, total virtualization of how we share and communicate, and then that personal contact and interactions. And I hope we can reach a balance.
And actually, you know, to build on that, Kimberly and Susan, would you have anything else to add about some of the lessons that we might be able to learn around COVID and thinking about how to take advantage of what's obviously an opportunity or a realization about the the ability of people to really come together to focus on urgency and need for a particular disease.
Yeah, I would just share that I have been absolutely overwhelmed and humbled by the opportunity and commitment and dedication of the community to find ways to come together. We have the meetup starting in July, but even before that, we have had so many of these collaborative opportunities and people show up. I think we still desperately, as a community, as a rare community, we need that connection. So I think it's been really, really powerful. I keep hearing so much about telemedicine, I think is what I would throw out there, just again and again and again, and how that will play out as things go forward. And there are some, some policy and some payer issues around that, but I think we can all work together to address it. Susan?
I would just say, to quote one of our panelists, Catherine Beaverson, who was quoting one of her favorite quotes or authors, that when fishermen can't go out to sea, they mend their nets. And I think that's a great visual for our scenario right now, because yes, things have been very difficult over the past several months for all kinds of issues affecting the entire world. But what did the rare disease community do? They got busy. They tried to find ways to keep access to care consistent and to keep their research moving forward. They did things that they wouldn't normally be able to do, but they just kept moving forward. And I just think that's a testament to the resiliency of this community, and it was an honor to watch.
Thank you. And I think we have time for one more question. And this is an interesting one, kind of thinking about some of the developments around a little bit more of individualized approaches. And so Jim, there's a question that asks, what opportunities are there for standardization of some elements of drug development? For example, thinking about what we might learn from one gene therapy vector and be able to apply to other diseases or other indications in order to ease and facilitate the development for some ultra rare conditions.
Yeah, I mean, that's an excellent question, and one that can be realized when it's built on a platform. So if there's a core component of a product that has been used in a previous product, we should be able to leverage that in a very significant way to simplify and ease the preclinical data that would need to be generated to de-risk the program, and actually some aspects of the clinical program. And that's the one thing that's marvelous about platforms. If you know how to produce AAV9 for spinal muscular atrophy type 1, it is the same manufacturing process for every other application of AAV9. So there's enormous opportunity for this, and it does require cooperation in terms of sharing information, but the regulators are very aggressive in trying to encourage that with the sponsors.
Wonderful, thank you so much. Well, I just want to take an opportunity as well to thank everybody. And Susan, I'll turn it back over to you because I think we're just about ready to start with our keynote.
Okay, so as I tried to say before, it's my very favorite part. I have the privilege of introducing our keynote speaker, Chris Austin, who will be speaking about innovations and collaborations advancing translational research for rare diseases. Dr. Austin is the director of the National Center for Advancing Translational Sciences, also known as NCATS, of the National Institutes of Health, which is responsible for several programs and initiatives for rare disease research and therapy development. Dr. Austin has founded several initiatives, including the NIH Chemical Genomics Center, the Therapeutics for Rare and Neglected Diseases program, and the Toxicology in the 21st Century program. He earned a bachelor's degree from Princeton University and an MD from Harvard Medical School. So we're thrilled to welcome Chris.
And I do apologize. I just need to add him here. The panelists, my apologies for just one.
That's better.
Hi, Chris.
I was wondering when that might happen, so it's great to be with you. Thank you. So I am going to now try to share my screen. So let's see what happens. Here we go. So let me know if you can see that.
Yes, we can.
All right, so you can see that. Okay, so I've been listening since the very beginning, and I'm actually really glad you all got through your introductory remarks sooner, because I found the question and answer session really illuminating and on point for both the challenges and the opportunities that we have ahead, and you'll hear some of the themes that you and Jim spoke about coming up here in what I'm going to tell you. I was really challenged to decide what to tell you about, because there is so much going on in the rare disease world at NCATS and elsewhere, at UPenn as a prominent example led by Jim. And a number of the most exciting things I actually can't tell you about because they're not public yet, but they will be very shortly, and I will try to hint at them, give you a nod and a wink whenever I can. And I'm glad to perhaps speak about some of those if some of you have questions about them afterwards. And you should be hearing about a number of these new things probably within the next 3 or 4 months. Okay, so I always like to start with this sort of view of the world, because it's really how I view the world, that this is the time in which we live. And all of you who fight the rare disease fight are very well aware of this, that over the last 30 or 40 years of my professional career and many of us, fundamental science has been absolutely transformed. And 2 examples, of course, are the Human Genome Project and what's pictured in that picture in the lower left, which is a group of stem cells, induced pluripotent stem cells, differentiating into nerve cells and putting out little processes there. I mean, when I was in training in the late 80s, early 90s, all of this stuff was really belonged on Star Trek, which I was a big fan of, I must tell you, growing up. But it's a reality now. These are things that first-year graduate students use now. But if you ask, well, from a clinical side, that's on the right side, how we doing? Have things really changed? Have they changed to the epical degree that science has changed over the last 30, 40 years? I'm afraid the answer has to be no. If you think about the last time each of us went to see a doctor, how different was that than it was 30 years ago, for those of you who were alive 30 years ago? The answer is not much. I'm a neurologist by training, and the vast majority of the diseases that I took care of and could not treat 35 years ago, I take care of and cannot treat today, even though we understand them sometimes in exquisite detail. So this is the problem we have to solve, and this is the translational problem. Translation is not new. People have been trying to do translation for thousands of years. The difference is we know so much more about the underlying science than we did before. And to the degree that this what we all were taught has not happened, and that what I'm talking about is, we were all taught that, gosh, if we understand the fundamental basis of human functioning and health and disease, we will, as the night follows the day, have treatments for them. That's what I was taught by implication, if not directly. And yet that has not happened. So we have to ask ourselves, why? Why is that? Why has this yawning gap opened up, and what are we going to do about it? And that's what I'm going to tell you about. Okay, so one of the ways that I think about this, I'm a geneticist, model organism geneticist, and a human geneticist by training on the basic side, in addition to being a clinical neurologist. And so I think about genes, and I know all of you on the call, particularly because one of the sponsors is global genes. I know you think about genes a lot. And I always like to say, when I studied for the boards in the mid-'80s, it was pretty easy because you could see the number of human conditions, the molecular genetic basis of which was known. There weren't that many. So, but now, during my professional lifetime, the number of human conditions, most of which are rare diseases, that have known molecular basis or genetic basis has gone from a few dozen to well over 6,000. But, and that's wonderful. And it's allowed diagnoses to be made at a level of precision which was simply impossible before, and to understand diseases at a level which was impossible before. But of course, what every patient wants is not just a diagnosis. A name is very salutary. I don't want to minimize that. But this is what you really want. You want a treatment. And the sad fact is, as you all know, 95% of these have no treatment. And I did a calculation a few years ago. At the current rate of diseases, and again, numerically, most of these are rare diseases, going from the untreatable, and by untreatable, I mean a specific treatment approved by a regulatory agency, in this country, FDA. That's what I mean by treatment. A specific treatment approved by a regulatory agency at the rate of those untreatable diseases becoming treatable every year. How many years will it be at the current rate before each of these is treatable? And the answer, unfortunately, is over 2,000 years. 2,000 years. So Global Genes is going to be having an annual meeting 1,999 years from now, and they will be showing this graph again, and we still will not be to the top. Now, to me, all you need to know is that number to tell you we cannot continue doing what we're doing and expect a different result. Science is progressing. We've seen that. But translation is not progressing anywhere nearly as fast as it has to, and it can. And I often say, you know, when Jim and I were in training, and we actually trained at some of the same places, you know, as a neurologist, I could sleep at night because I couldn't treat most of my patients, but I didn't know what caused their diseases. I can't say that anymore. I know what causes them. I know what to do. But the system we have has not evolved to allow that to happen. It's still, to a great degree, in opposition to actually what needs to be done. So there's this old adage that you all know, that what got us here won't get us there. And that's the situation that we're in now. And you all on the phone, as you heard from Jim, you all on the phone are a key disruptive technology to make that happen. You may not have been called a disruptive technology before, but you are. Okay, so why is this? The fact is that during this amazing boom of technological innovation and knowledge, our efficiency of developing therapeutics has consistently declined. So those of you who know anything about economics know about the concept of productivity. That is, that you would be able to make $1 or $100 gets you more of an item over years, and that's that basis of economic productivity is what allows companies to grow and economies to grow, et cetera, et cetera. What you're looking at here is negative productivity growth of therapeutic development, monotonically negative productivity growth over the last 60 years. So what that means is, sort of bottom line here, the number of new drugs approved by the FDA has gone down by half every 9 years since 1950 per $1 billion spent. And you ask, well, what would happen in a circumstance like this? And I don't care whether you're talking about drugs or shoes or ball bearings or cars. If your efficiency of making your product, or that is the price of your product, doubles per output every year since 1950, what is going to happen? Your companies are going to go out of business because they're not going to be able to afford to stay in business. Companies are going to merge with each other, and the products that they do make are going to be exorbitantly expensive. So I just described the pharmaceutical industry for you. So this is not a surprise. It's a matter of non-simple mathematics. So what's the answer to this? The answer, in our view, is science. I often say that we're in the same situation as Matt Damon was in The Martian. movie. You remember I hope you all saw that movie, where he's also in a foreign environment where lots of people have died and the trend line is not good. And what does he say? There's this critical moment in the movie where he says, I can't say what he really said because this is a family show, but he says, I am going to science the stuffing out of this. And that's what he does. And that's the answer here too. The translational process, for a variety of reasons, historical, cultural reasons, is an almost relentlessly empirical trial and error process. And so if you have a process that increases in complexity with so many orders of magnitude, if you let me use that word, over the course of about 20 years, a 20-step process, which becomes more and more and more complicated as you go from beginning to end. If you have a trial and error approach, you don't know what you're doing, so you're essentially taking guesses, some more educated than others. What is the likelihood of you're getting from the beginning to the end? It approaches zero. And that's exactly what you see. That's why we have these numbers of only 1 out of 10,000 compounds actually ever gets approved. It's all true. But the answer to this is not shots on goal. I'm sure you've all heard that. That drives me nuts. If you have a 1 in 10,000 chance of something working and you double the output, what do you get? 1 in 5,000. That, my friends, is not something to celebrate. We need to have 1 in 100. We need to work on order of magnitude improvements. And science is the only way we're going to allow this. So in other words, what I was taught by very well-meaning people, this is what a neurologist and a geneticist does all day. We make diagnoses for diseases we can't treat. That's always the saw on neurologists. And we publish papers of genetic causes of diseases. This happens to be the first molecular disease, sickle cell anemia, 1949, is not the equivalent of having a drug or a device. And furthermore, having a drug or a device is not the same as access to that drug or the device and health of the people who you diagnosed way back at the beginning. So these two inequalities are the translational problem. How do we make them as close to equality as we can? And the current rate, we're one might say we got nowhere to go but up. These are the current success rates. It's 30 years for an individual intervention for an individual disease, But one of the primary reasons, if you ask yourself, so I realize you may be entering terminal depression here, but please don't, because I'm going to rescue you, because I do think the answer is science. But a lot of this, I must say, is it's a cultural thing. So when I had this memorable conversation with a mom of a rare disease patient a number of years ago, and we were discussing this problem of the disease, the research enterprise's love affair with fundamental discovery alone, without translation. And she said, I love basic discovery. I love papers. I love publications. But the fact is, when my daughter gets sick, I can't give her a publication. And I think of that almost every day, because that, to me, crystallizes the problem. And don't get me wrong, for us nerds, people like Jim and me who live in those inequalities, there is amazing science to do in this space. But you need to know that the traditional academic model doesn't accept that. It's, science is defined very narrowly, and that's one of the problems that we need to confront. Okay, so what is this thing called translation? People throw this term around, and it's a terrible term because it's got lots of other meanings, of course. But really, what you need to understand about translation, it is the term of art for turning an observation into an intervention that is shown to improve human health. And it's very broad. It's modality independent. It's small molecule, biologic, and antibody, and gene therapy, behavioral intervention, et cetera. But this process of going from an observation a scientist makes, or a patient makes, or a parent makes on a disease, a doctor makes for the first time, when a light bulb goes on and says, gosh, if I could just reverse this thing, you know, I might be able to reverse this problem in front of me. You know, that whole process of creating the thing, you know, the gene therapy vector, the small molecule that actually does what you want it to, and getting it to all the people who need it, and showing that they actually get better when they take it. That whole process is translation. And we use the term translational research, and excuse me for the, pardon the lexicology here, but these words, we have to, I always like to define how I use these words because people use them differently. Translational research, we define as the endeavor to traverse a particular step of this complex 20-step process or more for a particular target or a particular disease. And every institute at the NIH does translational research. Every biotech, every pharma, almost none do translational science. So what is translational science? It's like any other science. It's a field of investigation that seeks to understand what the general scientific and operational principles are that underlie this process, with the thesis that if we can do this, we will transform translation from being what it is now, which is an empirical trial and error phenomenological process, into a predictive science. It will go from being what I sometimes feel as a practitioner of this for my entire career. So I am well suited to say this. My field often feels more like astrology than science. But we will transform it from astrology to engineering. That's what we need to do. That will increase productivity, that will decrease costs, that will decrease failure, that will increase success rate. But amazingly, very, very few people and very, very few organizations in the world do this. It really only started as a field when NCATS was formed almost 10 years ago. Okay, so what's the mission? And you're going to hear a lot about this over the next 2 days. You know, the rare disease world is full of this. New ways of doing things, new methods, new technologies, new paradigms, new ways for new collaborative paradigms, new public-private paradigms, ways for people to work together. And that's what NCATS is all about. So one of the things that we work on, we actually made a list of the major causes of translational failure a number of years ago now. And the important thing for you to know is it doesn't matter what your disease is, the problems are all the same. They're all the same. Doesn't matter what disease you work on. They are all variations on a theme. These are the things that kill translational projects very effectively. And we work on all of these. I, of course, don't have time to go through all of these. I'm glad to give a 1-hour talk on each of these subjects at any time, but I will not do that today, just to say that we know what the rate limiting steps are. That means the ones that we need to work on to develop ways to do it better, to improve the overall yield at the end. Okay, so this meeting is about drug development, so I thought I needed to show you, because of the keynote, I needed to show you, for those of you who don't think about this every day, you know, how do you do this? And what I'm showing you here is a little specific to the small molecule world. It's a little different for gene therapy and other things, but in general, the idea is the same. There's basic research that we all know and love, but that's not translation. Developing an assay, that means a testing system in order to test your intervention in cells, and then in multicellular systems like what you've probably heard of, organoids or tissue chips, and eventually in animals. After we create that testing system, then we often need to screen lots of stuff. And you know, this is actually a picture of one of our robots. We love robots, but think about the fact that we have to use robots to find drugs. The fact that we have to test 3 million compounds to find the starting point of a new drug is tacit admission. We have no idea what we're looking for, right? I mean, it's very impressive tour de force, but I want to put these robots out of business. I mean, if somebody came to me with a new rare disease, I would love to be able to say, I know what's going to treat that. I don't have to throw 3 million random guesses at this. So trying to figure out what the rules are is what we're trying to do. And very few, if anybody else, does this. It's really quite interesting. It has to do with motivations and incentives and all kinds of things. But then virtually never, except in the case of repurposing, do you get actually a drug out. We do a lot of repurposing, but particularly for rare diseases, we're getting into novel targets, novel parts of genome space. It often doesn't yield what we really want. So often we need to start with a new chemical entity, modify it for human use. That takes 3-4 years, testing in animals or other systems, and then eventually clinical development. And NCATS works across this spectrum. And then, as you probably know, in the clinical world, then you have to start over, and you have to go to multiple phases of clinical trials. And often people think, well, gosh, FDA approval happens if it needs to happen. Behavior, it doesn't. Behavioral intervention, of course. And FDA approval is after that. But really, in many ways, that's when translation begins, because you have to assess its effectiveness in the real world of medical practice settings and then in public health. And so NCATS has programs across these as well. And I want to emphasize what's on the left there, and you heard Jim talk about it. Without natural history and registries to know what the course of a disease is, you have no idea how to measure the impact, the salutary impact or the negative impact of an intervention you do. And that's, most of you will recognize that pink figure on the bottom there. Those are growth charts that all of you who have kids know what I'm talking about. And what did you do there? You plotted your kids' height and weight on against one of those things. You're doing natural history. You are doing a natural history study versus what is known to be the natural history of growth in populations. So if your child is either above or below, you know it. That's natural history. And we need to have that for every disease. And without that, we are really shooting blind. And I got to tell you, I've tried to fund a lot of these as even running a center. I've tried to fund these. I know Jim has, and many of us have. It's difficult in the academic world for reasons that you might want to discuss. Okay, so at NCATS, we have the privilege, and I say the rare privilege, of being the epicenter for rare disease research at the NIH. Many of you, if you've been around a long time, know Steve Groft, who was the original director of the Office of Rare Diseases, which then became the Office of Rare Diseases Research. It got incorporated into NCATS when NCATS got formed back in 2011, run by Ann Pariser. If you don't know Ann, you really need to get to know her. She is a force of nature, like a lot of you on the phone, I must say. And she is doing amazing things here. And I do not have time to tell you about all the things that ORDR is doing. But they do fit into roughly 3 categories. One is knowledge, gathering knowledge and sharing knowledge, and particularly integrative knowledge, taking knowledge of many, eventually we hope all rare diseases, and putting them into an environment where we can compare and contrast the characteristics of various rare diseases to understand which ones are related to which. Not only genetically, but by symptomatology and effects of drugs, et cetera. And then there's a lot of research, of course, and we have a lot of programs that we do. They are all collaborative programs. Everything NCATS does is a collaboration, everything. We're a very atypical institute for that reason. And then we do a lot of community engagement. And to us, interacting with and working with groups like Global Genes, and the Penn Center, I must say, because both are our communities, academic communities and patient communities, can't believe I'm going to say this to this group, but it is in our DNA. It's just what we do, and the reason is that we are very focused on what other people need, other parts of the translational ecosystem need. And the only way we can understand that is to engage with the communities that we hope to help. It's pretty fundamental. It's not very complicated, but it's pretty unusual in the scientific community, I'm afraid. Okay, so something that you need to know that I talk about at every opportunity, as does Ann, is that rare diseases as we like to say, rare diseases are not rare. You know, they are a public health issue. And until the public and our policymakers get their heads around this issue, we are never going to make the headway that we need to make. So what am I talking about here? You know this, but just to tell you the numbers that everyone, our policymakers certainly do not know, is that there are 7,000 of these. It never ceases to amaze me. Every time I say this, people fall out of their chairs. 7,000 diseases, I had no idea. 80% approximately are genetic. Most are childhood onset diseases. What you may not know, which is really quite interesting, is that about 250 new rare diseases are being identified every single year. And the rate of that is not decreasing. That is, how many rare diseases actually are there? There's a bit of a friendly competition in our field going on now about how many, where will we finally end up? Now, of course, it depends on how you define a disease, of course. But these are, by diseases here, I mean a new syndrome that might come out of the undiagnosed disease program or something like that, or a new relationship between a genetic mutation and a syndrome, a phenotype. So it's sometimes called a genotype-phenotype correlation. So that's what I mean here. Really interesting. So that 2,000 number that I gave you a few minutes ago, that's with 7,000 diseases. But every year, the goalposts change. So this even more reason to change the way we think about things. Okay, so and again, you know this. Most of your policymakers do not, but they are individually rare, of course, by definition, but they are cumulatively common. The total prevalence is approximately the same as diabetes. Now, how many people in the public sphere and your friends know about diabetes? Virtually everybody. How many people know about rare diseases? Frankly, almost nobody. How many people know that diabetes is a big problem? Virtually everybody. How many people know that rare diseases are a big problem? Virtually nobody. Rare diseases are a much bigger cause of loss of life, loss of productivity, loss of health, if not life, than diabetes is. And yet this is not understood. And we can come back to why and what we're doing about this, because we really are doing a lot about it. But we as a community really need to double down on this. Of course, very high costs in direct patient care, loss of productivity. You all know this and live it every day. You know about the diagnostic odyssey. That feeds into these high costs. And we talked about this 2,000 years before we have treatments. So what is the solution to this? At least one solution, in addition to understanding the translational process better from a scientific point of view, is to transition from a one disease at a time, which is how we've always done this, to a many diseases at a time approach. And what I mean by that is looking for commonalities among diseases, either at a genetic level, a cellular level, a symptom level, and working on them together so that you can compare and contrast and get insights very rapidly from one to the other. And what are called platform technologies. Whoever asked for diagnosis and treatment, whoever asked Jim about the AAV issue, AAV9, thanks to Jim and others, is now a platform technology. Whole genome sequencing is a platform technology. It's those kinds of things that we're looking at here. Okay, so we're doing a lot of things in this arena, providing information and big data through GUARD, which I'll tell you about in a second, empowering patients as research partners via the toolkit, developing interoperable registries via the RADAR program, and doing natural history and interventional studies via the Rare Disease Clinical Research Network across the NIH. We're also really trying to change perceptions. We're in the second or third round now of this Rare Diseases Are Not Rare challenge. This was first place from the first time we did this a couple of years ago. I love this one. If you can read this, more people know about unicorns than rare diseases. Yeah, I mean, this is what I love about this. This was a $5,000 prize, and you had to do something that was a visual something, you know, a poster or a video or a poem or something. And we got the most amazing proposals. submissions. The other is that we're partnering with other groups like yourself, with FDA, with patient groups. Rare Disease Day, you all know about that. So GARD, I hope you all know about GARD. It is actually a congressionally mandated information center. It's that is on our website. If you just look at GARD, it's one of the real probably the best in my mind patient-focused source of information. There are others that are focused on researchers like OMIM and OrphanNet. OrphanNet somewhere in between. It's in the European database, but this is unapologetically and intentionally patient-parent focused. And in addition to having all kinds of information about what the disease is and what the causes are and how you can get help and foundations that you can connect with, there's also somebody on the other end of that phone, because every parent that I know who's gotten a diagnosis wants to talk to somebody. And so if you can't figure it out, you can pick up the phone and call somebody, and we employ folks who will talk to you about how to find the information you're looking for. And this is the example. I just picked one of thousands, 7,000 Ehlers-Danlos syndrome. But the point I want to make here is that it's if you look at the language there, it's all it's not nerd speak, it's not science speak, it's normal people speak, except for a few things like autosomal dominant. And when we use a term like that, it's underlined, which means it's defined. So I hope you'll take a look at that if you haven't seen it so far. The toolkit for patient-focused drug development, I certainly hope you've had a look at that to help with your own work. A lot of work was done with a number of patient groups. We developed this with a number of patient groups. The two people who really helped run it are two people, a lot of you know, Annie Kennedy and Ron Bartek. They were the two people. There's a big group who helped us with this. And a lot of your Global Genes resources are on this website. And also the Rare Disease Registry program. This is not a program where a platform where you could do a registry, but it's what do you need to do if you want to start a registry? How do you do it right so that it's comparable as much as possible with other registries, and it is done in a way that FDA will like, and that's really important. Let me just see. Okay. So I want to get to a couple of things specific here that I think you'll find particularly exciting. This is a new thing. It's actually something that we've been working on for 2 years, but I can now talk about it. And it relates directly to this question of how we democratize gene therapy. I think we've all been impressed by examples of how monogenic diseases, some monogenic diseases have really dramatic effects given gene therapy. And AAV vectors, has been pointed out, have become a delivery mechanism of choice for these. But the problem is the current approach to gene therapy is still one disease at a time. A lot of people and companies are competing on vectors. That is, it's completely the opposite, and you need to try to get your head around this. It's completely the opposite of a small molecule, where a company will have what's known composition of matter intellectual property on the actual agent that treats the disease. That is generally not the case in gene therapy. People are delivering, and I'm grossly generalizing here, they are delivering a wild type gene. That is not patented. So the only thing you can patent is the suitcase it goes into. So people are competing on the suitcases, not what's in them. That, in our minds, is understandable commercially, but silly medically. And so we really have knocked ourselves out trying to figure out, well, what could we do about this, particularly for the diseases that you were talking about before, that will have no commercial model. There are many of these disorders, and including maybe the ones that all of you have, that have a couple of dozen patients in the world at any one time, maybe 100. Just think about it. To do a properly powered clinical trial to get approval for the gene therapy, you will have to treat all patients currently living. That leaves no population to sell it to. So no company is ever going to develop that. So what do we do about it? So that's what we're working on. And so the idea is, could we design clinical trials on a platform level, multiple diseases at a time, using the same vector at the time? And so this is something called PageGT. This just went live, this site, so I hope you'll take a look at it. It goes into great detail about what PageGT is, how we're doing it. It's a collaboration between ourselves and a collaborator at NSGRI and a collaborator at NINDS. These are, in case you know these people, this is Chuck Venditti, who works on organic acidemias, and Karsten Bonneman, who works on a number of neurological diseases. These happen to be congenital myasthenias. But the plan is to develop in the same AV serotype 2 different genes that is the same suitcase, 4 different cargos in that suitcase for 2 different types of a similar disease, one at NHGRI and one on NINDS, and do all the safety, all the toxicology, get into people, do the clinical trials, and make all the data, all the manufacturing data, all the CMC data, all the clinical data public. Put it on our website, including all of the correspondence with the FDA. So that if a researcher, years from now, hopefully not too many years from now, has a new rare disease that affects 2 people or 100 people or 20 people, there will be a cookbook. If you follow this cookbook, FDA will not ask you to do any of the work that they normally do, because we've done it. And as long as you stay within these parameters, you can go directly into people as fast as possible. That's the idea. But we've also realized, and this will be absolutely transformational when that happens. And we're working with a lot of other people on this. Don't get me wrong, including folks like Jim and Jim. But we've become aware that this is, we have another roadblock, and it's a classic translational roadblock. What you're seeing here is the number of IND applications to the FDA for gene therapy over the last few years. And there are currently over 900 INDs for gene therapy as of the end of last year. And those of you who tried to get a gene therapy vector construct manufactured know that currently it is extraordinarily expensive and takes, and there's a 2-3 year waiting list now. And and it's clear, though, there is new building going on here. The market is responding. The problem is that the need is going up exponentially. That's what you're looking on the blue line, and the capacity is increasing linearly. You know, so let's say if we're doubling capacity every 3 or 4 years, and we're doubling number of trials every year, we are never going to catch up. We can't build our way out of this problem. We have to increase efficiency. Same old story. And so we've been working very hard with many of you in the community, and very closely with Peter Marks, who runs CBER, on more efficient, greater than 10-fold manufacturing technologies needed to meet demand. This is a workshop that we had that Jim and many others presented at last January, pre-COVID, about these problems. And there is a very big effort which is going to come out about this soon. I have to metaphorically shoot all of you if I tell you what it is, but I think you'll find it very exciting, and I certainly find it very exciting. So you hear about that in the next couple of months. Natural history and clinical trial networks, really critical. Natural history and registries that lead to natural history is often the big limiting factor in being able to effectively test an intervention. FDA really wants these data. They really want the data, particularly if you're proposing a non-placebo controlled trial. The only control group is natural history. So you just gotta have these data. This network, it's a great network. Don't get me wrong. There are many NIH partners. You may recognize some of your diseases here. The yellow ones are the ones that are new in this cycle. Started about 6 months ago. This is the 4th cycle that we've had here, but it still only studies about 300 diseases. That leaves 6,000 and change to go. But the important thing is that these consortia do not, first of all, they're all consortia. It's not single site. Each one of them has about 10 or 15 sites, and virtually all of them are international, have international sites too. But you can't just study a single disease. You have to look for a commonality. And different ones group in different ways. So you look at a urea cycle, right? That's all of the rare diseases that affect a particular biochemical pathway. Or you look at symptomatology related disease, ALS and related disorders, or other symptoms like dystonia, or an organelle in the cell, like mitochondria. But they have to study between at least 3 up to 40 different diseases. I think lysosomal storage diseases is the reigning champ of numbers that they try to study. And what we see over and over again is studying those related diseases is dramatically advances progress. The other thing that dramatically advances progress is the last bullet here. That is that patient advocacy group, you have to have at least one patient advocacy group on the research team to get funded. Not as a group that, you have coffee with once a year and get a check off, on the research team. And I don't think I need to tell you all why that's so important, but it really has been transformational for the progress, for the productivity of these groups. So I mentioned this, and it's another mantra for us. The patient membership on the research team, we think, is a disruptive technology for rare disease therapy development. We are approaching this in a science-driven way. As you all know, relationships and teams are, it's easy to say, hard to do. So we need to have a kind of a roadmap, and you all have done some of this, and we need to do it on the researcher side. For if you're going to do a collaborative project involving a number of rare disease groups and academia and government and FDA, how would you do it? How do you deal with, among other things, like data sharing? And that was what we did at NCAT's Day. That's a day we have every year to bring patients and patient advocacy groups in and talk to us about a particular issue. I want to just speak for a moment about what we've done to COVID-19. I don't want to minimize the disruption this has had to the research ecosystem. Most of our labs, as in most places, have been closed. Most trials that are not COVID trials have stopped. They're beginning to start again. But in order to get a handle on what exactly the impacts have been on rare disease patients and their families, to position us to restart in the most effective way, we decided we needed to do a survey to figure out what's actually going on. So, and we have been pushing here at NIH to get rare disease populations defined as a medically vulnerable population. Just like patients with respiratory diseases, et cetera, lots of rare disease patients, not all, but are medically vulnerable and have to be treated with great care when it comes to COVID, as you all know. So why are we doing this? People with rare diseases, and we've heard this loud and clear from all of you, are concerned about a lot of things: impact on their conditions, access to medications and support and research. And so we got together with our RDCRN, that's the Rare Disease Clinical Research Network, patient advocacy leaders, something called the Coalition of Patient Advocacy Groups, or CPAG, and the PIs of the RDCRN centers, to figure out a consistent way to gather information from the community and help us, as we say at the bottom here, plan for gaps and needs and alternative strategies. These are the things we're trying to learn. What are the concerns of rare disease patients and their caregivers? Estimate the number of rare disease patients who actually got COVID-19. Was it more or less? I'm guessing it's less, because you probably all quarantined yourselves and didn't go out, but we'll see. Describe character, if there are differences in COVID-19 symptoms in rare disease patients, subgroups, possible interactions, hoping to enroll 5,000 people or more. And I hope you will take the survey if you have not already. We're aiming for 5,000 people. We're almost at 3,000 now. But we really need as many people as possible. Data are only as good as they are abundant and representative. It takes about 20 minutes to complete. It's very simple. The easiest way to find it, you can go to that long URL there, or if you just Google RDCRN COVID, you'll find it. I looked for it again this morning. So please do consider doing that. And if you have any questions, we do have two of our program leads, Tina Erb and Mira Shah. You'll have these slides, so you should feel free to contact them. Okay, just real quickly, before we finish up here and go to questions, I wanted to get across to you what your opportunity is to work with us specifically. And lots of organizations that you'll hear from are collaborative organizations, but I wanted to give you a sense of not only the resources and the research that we're doing, but how you work with us if you want to. So we have investigators and disease advocacy groups come to us at various stages of this process, and they get stuck at one or more of these purple arrows in the preclinical space, and they when they apply and they get in, then they get slotted into one or another of these programs that I'm not going to talk about, but just know that there are many programs that these go into, many of them focused exclusively on rare diseases, as you can see from trend there. And then we'll come out the bottom are deliverables that are useful for that investigator or that rare disease to move forward down the translational pipeline, but they also teach us something about the translational process. And just one of the branches that I thought might be most interesting is the one that's between what's called lead optimization and IND. This is run by Don Lowe, and I've given you his URL there. Does IND enabling studies starting with a lead. These are very expensive, very hard to get funded. Commercial investment is very scarce. This is a crossing the valley of death enterprise. This program lives in the valley of death very proudly. So contact Don if you have questions about that. We're doing a lot to try to improve the predictivity of the translational process. And one of the things we're trying to do is to mimic human tissues and cells in a more faithful way than we could do 10, 20, 30 years ago. There are lots of technologies now with increasing complexity going all the way to an organ on a chip. The issue and the way to think about these is that they're all useful. They just have different levels of throughput. And the more complex you get, the lower the throughput is. So on the left side, we can do 3 million of those on the left where it says 2D cells in a dish. We can do 3 million of those in a week. Organ on a chip, you can do maybe 10 a week. So they're both, but they're both very useful. If you haven't caught up with the tissue chip revolution, please do. This has all happened in the last 8 or 10 years. NCATS has led and done most of this work, I'm happy to say. The idea here is instead of trying to study a disease in a rat or a mouse engineered to have a human's gene or gene mutation, which, don't get me wrong, can be very useful, but mice are mice and humans are humans. And so could we develop multicellular bioreactors in a microfluidic platform that would represent the structure and function of all human tissues, and use those to understand the disease and test the safety and effectiveness of new therapeutics. And this was a pretty crazy idea 10 years ago. It's a reality today. We have about 14 different organs. We've actually hooked them all together in certain circumstances. And I just want to show you that we've started using these to study diseases as well. And the ones in red are all rare diseases. The ones in blue are common diseases. And you'll recognize a lot of these rare disease names. But this is really something that you want to look into if you haven't already. So finally, I want to just give you some other resources. If you're trying to figure out, in addition to the roadmap and to the toolkit, which I hope you'll find useful, if you're trying to figure out what the heck all these people, these researchers and these drug companies are talking about when it comes to translation, just give me a freaking map so I know where I am. We realized a few years ago that nobody ever created such a map. Getting together with Sharon Terry and some of her colleagues at Genetic Alliance, some of you may know that they created I actually helped her with this, a very early version of this. We brought together a group of people from academia, from pharma, from regulatory agencies via a process of the National Academy of Medicine to develop such a map, and we published them a number of years ago. And the point I want to make, and this is all available to you freely on our website, and I'll show you that. But this is the chevron that I've shown you, and you've seen 1,000 times. This makes it look really easy, right? It's just, it's drug development in 6 easy steps. What could be harder, right? I mean, it's a pipeline. How hard can it be, right? Well, this actually isn't what it looks like. So we decided the bad news is, we had to let people know, the bad news is it looks like this. And this is actually a low mag view. This is a greatly simplified view. But this is what it actually takes to go from a discovery to a therapeutic that reaches patients. But like anything, if you're going to start on a voyage, you gotta have a map and you gotta know where you're going. And then we realized, well, gosh, static maps in kind of 20th century, we really need a Google Map version. So we created a Google Map version that's on our website. You can zoom in, you can zoom out, you can link to resources. This thing currently has 10,000 nodes in it. That is 10,000 different steps and different organizations that are involved in this. This really is how complicated it is. But how do you make it understandable, and you're only interested in one part of this? Please go here and you'll notice the word patient there in the middle. So you're very much part of this process. And lastly, this is really for the nerds in the group. And by nerd, I want you to know that that's a term of endearment for me. I'm a proud nerd myself. One of the problems here for investigators getting into the first time, there really is not a literature of this, because everything we're talking about has pretty much not been an academic discipline. So there is very little literature on a how-to. So we created this with initially colleagues at Eli Lilly, the big drug company, but it now has an international editorial board. It's updated regularly, and it's free. So some of you, I know Jim will remember this. This is how I learned how to clone some we all call maniatis, and then something called current protocols of molecular biology more recently. These are subscriptions or books you have to buy, and they're cookbooks. That's what they are. They're pure and simple. They're cookbooks. But these are the cookbooks that, like one of those good Julia Child things, it tells you what the secrets are. What do you got to know to make this work? And so they're detailed protocols. It's now about 1,400 pages, about 50 different chapters. So if you have collaborators, investigators you're working with, please point them at this. It's all free, and please do take advantage of it. So I know I've given you a lot of information, and I know I started out with some pretty daunting numbers, and I did that on purpose because I wanted to challenge you to continue doing what you're doing. which is to argue for change in the system and to be an active player in that change. This is the NCAT's translational science spectrum diagram that we show everywhere. This is how we think about translation, going clockwise or counterclockwise. But the point I want to make here is look who we put right in the middle. And we did this for a lot of reasons. One is that our constituents are the patients. I love my colleagues in science, but sometimes we forget that, our grantees, our investigators are not who we report to. I mean, we love our grantees, but only to the degree that they do science, which in our hands advances science and advances the health of patients. So we put patients right in the middle of everything we do. And we find that when we do that, some of the decisions that we have to make actually become relatively simple. I've been in a number of meetings with my staff where I say, okay, this is a really hard problem. If you were a parent or a patient of a rare disease, what would you want? What's the decision that you would like us to make? And as soon as we ask it that way, the decision becomes quite simple, actually. So thank you for all you do. I heard Jim's first point. I think he said something about we need you. We're not kidding. We are not being patronizing here. Believe me, we need you as members of the team. We can't do this without you. And so please engage with us. If we're not doing something the way we need to, please tell us. I think any good relationship. And I'd certainly be glad to take any questions for any time that we have remaining.
Thank you so much, Chris. That was tremendous. Covered so much ground, as you said. Thank you for educating us on the translational science spectrum, reminding us about the painfully slow pace of identifying treatments and the need to increase efficiency, how patient membership on the research team is disruptive technology. That was fantastic. The importance of defining the rare disease community as medically vulnerable, very important. Thank you for sharing your patient survey with us. Actually, Global Genes is promoting that too, so we're gonna help you get those, those respondents. If you can't find it on the NIH website, you can find it on ours. We're working with Dr. Macaluso on that. Chip's fascinating science, and thank you on behalf of everyone for literally putting patients at the center.
Yeah.
We do have a series of questions. I'm actually going to open with the one that we actually posed to Jim earlier, and want to give you a chance to respond as well. So we've seen unprecedented collaboration among academic and pharma company researchers in the midst of COVID, right?
Right.
How do we take advantage of this precedence across rare diseases? What do you think?
Yeah, like a lot of you, I'm thinking a lot about this now. A lot of things which are happening now are things that I have been advocating for sometimes decades. And I've known all along that they're possible. And I've been getting lame excuses about why various groups couldn't do something. I couldn't share, I couldn't do this, I couldn't do that. And we've seen that fallacy come falling down very rapidly. Because as you say, almost overnight, the entire research ecosystem started working together as a single team. And what you've seen is incredible progress, to the degree that the public is now worried about the safety of these drugs, which I understand because this is being developed so much faster, and they think that shortcuts must have been taken. I can tell you, shortcuts are not being taken. It's just all the stupid, duplicative secrecy and tribalism and siloism that has gone away, and that is to be celebrated. And it shows you how much more efficiency we can get if we just act the right way, and we could do it tomorrow. So I've become fond of saying, this genie is out of the bottle. People can't say they can't do it anymore. They've done it. So now it's clearly, it's an I won't. So you need, and we need to put the question to them. So let me get this straight. You're willing to do this for COVID, but you're not willing to do it with this child with a rare disease? Are you kidding me? Really? And challenge them that way. And the pharmas and the FDA and NIH were all saying the right thing, but you got to hold us to it. You got to hold us to it. We're going to try, but as the research community, any community, has a great sense of regression to previous operating. So I'm become fond of saying we're trying to figure out how to get a cork in the bottle before the genie gets back in.
Right.
But I think the other thing to mention, which I think is a real learning opportunity here, those of you on the phone are very well aware of the urgency that you all feel. And one of the frustrations I know you feel, and I feel it every day too, is that most of our fellow citizens do not feel that. And the reason they don't feel that is that they haven't had a disease yet. They haven't had a child born with a rare disease yet. They haven't had a diagnosis themselves yet. Overnight, when that happens, they are transformed. They are evangelists. But most of the population says it doesn't apply to me. Here, everybody realizes the urgency. They know it. And so we need to capture that sense of urgency, which everyone knows now for the first time, most of them, and say, there are 25 million people in this country who are being, if I could be a little exaggerating just for a moment, just a teeny bit, ignored, when some of these diseases have greater morbidity and mortality, greater decrement in life and quality in life than COVID does. So what's the deal here, people? Society. So I think it's, so the community has learned that they can do this and the world will not come to an end. They've learned that that productivity really does skyrocket, and that's a good thing. And we need to, and you need to play a big role in this in preventing us from going back to our old ways.
Right. And somebody asked, you talk about awareness and how everybody knows about diabetes, but not about rare diseases. Someone asks if we change the nomenclature of the naming system of rare diseases, do you think it would increase public awareness? Think we need to
boy, yeah, we thought about that quite a bit. We thought about rare. We've actually had whole focus groups and all kinds of things, and we thought in the end we couldn't come up with a better name, and then we thought, well, gosh, Nord would kill us if we changed the name, because this is, the word rare is enshrined not only in organizations, but in legislation and all kinds, the Office of Rare Diseases, right? So
yeah.
So that's why we decided, but I take your point. That's why we've decided to go with the slogan that rare diseases are not rare, and to change in people's view, including how each of us speaks about diseases, that we speak about them as rare diseases first and the specific disease second. We have never done that. So the public, and I can tell you Congress, does not understand that these are all one big group called rare diseases. Now, cancer does not have that problem. If you ask somebody who has a leiomyosarcoma, what do they have? They don't say leiomyosarcoma, they say I have cancer. And then you ask them, well, what kind? Well, I have a leiomyosarcoma. So the Cancer Institute, as you may have noticed, has a slightly larger budget than the Office of Rare Diseases. But that's because policymakers understand the public health significance. It's the same reason that the Alzheimer budget has quadrupled in the last 4 years. It's not because people in Congress all of a sudden realize this is a big health problem. They realize that this is an advocacy issue and this is an economic issue. And that's really what drove it.
Yeah. So tell us, Chris, how has COVID disrupted your research at NIH and NCAP? And when do you anticipate the disruption to end? How can
Well,
patients
we've been required. The labs were all closed the second week in March, and all of us who could were have been told to telecommute. I happen to be in my office today because I didn't trust the technology, but I've been in my office a couple of times in the last 3 months, 3.5 months. The labs have all closed. We do have about 10% of the staff working on COVID drug development and assays and screening and all of that, and clinical trials. We are going to be opening up the labs again, we hope, a week from Monday. The clinical trials for rare diseases, recruitment and visits have essentially stopped, including at the NIH Clinical Center. The only visits that have been allowed are for existing patients on critical cancer protocols. And I think what remains to be seen is a really interesting question from a public health point of view. That is, will we find that there is more suffering and death from the indirect effects of COVID on all the other diseases, given what's gone on in the last 3 months, than morbidity and mortality from COVID itself? I mean, you've probably seen the data on heart attacks and strokes and all that. People are not going to the hospital, and if they're not getting treated, they're dying. Now, those aren't listed as COVID mortality statistics, right? But they may actually, I think in the end, and rare diseases fit in this too, they may actually exceed the number of people who were affected by COVID. So it's a scary thought. It's a scary thought.
Yeah. So one question from an industry perspective and one from a patient perspective. Practically speaking, how can biotech work with NCATS in order to accelerate drug development for rare diseases?
It's a really important question. So, I talked about community engagement. Academics are certainly one of our communities. Patients are certainly one of our communities. But biotech and pharma and VC are equally one of our communities. So we work very closely with them. You should know that our board, it's called our council, but it's essentially a board of directors. A third of those people are from biotech and pharma and VC. A third of them are patients and patient advocates, and a third are everybody else. That preclinical, that therapeutic development branch slide I showed you, about half of those collaborations are with biotechs and pharmas who have projects on products and diseases which are not commercially viable. So we form a joint project team with them. The funding comes from us. For them, it's in finance speak, it's non-dilutive capital for them, and it advances our mission because we advance this down the pipeline enough to an IND or maybe first in human clinical trials, and then it graduates, and the company can either go on its own or be acquired. One recent example you may have seen, we partnered with a company called Agilis a number of years ago. It's a gene therapy company in Boston working on AADC deficiency, and we got all the way to the point of a BLA, and at that point PTC Therapeutics came in and bought Agilis and is completing the clinical development, and that's a very common issue. So we're very proud of that function, and we do a lot of it. But you should also know that on most of the project teams that we have, including Tissue Chips and others, there are industry people on those groups.
Okay, very good. So then from a patient, she says, there is a big gap between patient-reported symptoms, disease phenotype, and what is reported in literature, affecting translational research and definitely affecting clinical care. Any recommendations, Chris, that you have for patient communities in helping to bridge the gap between what's published in scientific literature regarding specific disease symptoms and then what's observed in the patient community?
Yeah. It's a complicated question, but let me give you a couple of couple of thoughts. So first of all, you should know a secret of medical education, that medical education is based on triads. That is, when you are being taught in medical school about something, there's always a triad. There's 3 symptoms that define this. So if you have appendicitis, it's a high white count, fever, and right lower quadrant pain. With a rare disease patient, it's going to be 1, 2, and 3. Now, that rare disease patient may have 10 other symptoms, and they're going to vary, of course, as a phenotypic spectrum. But you can't keep all those straight. So all you can remember is that there's these 3, and that's how it's defined. And everything else goes by the wayside. To some degree, it's just a practical human cognition issue. Now, of course, that's not limiting anymore, because we have these things called computers and databases, which we didn't have back in the old days, like when I trained. And so we're needing to redefine the phenotypic spectrum, that's what it's called, via databases like GARD. And the ultimate solution to this, an ultimate solution, would be an Icd code. So Icd codes and lack thereof are an enormous problem in the rare disease community. You probably know that in the Icd-9, there were only about 500 rare diseases that had had a code, and in Icd-10, it's maybe up to about 2,000 at best. And those are international classifications of diseases, and they list symptoms, and they're actually very helpful. But for most rare diseases, those don't exist. And part of it is that there is not enough experience of a broad enough group of people to be able to define the phenotypic spectrum that patients know exist and documented in the medical literature. So this brings us back to registries in natural history. The only way we're going to define this is by gathering the patient experience, and that is in a natural history study, a rigorously conducted research study which looks at all potential, all symptoms that a group of patients have. And of course, not all patients have the same symptom of any disease. You've probably seen this about COVID, right? It's percentage of diseases. Not everybody has cough. Not everybody has fever, and yet they have COVID. It's the same thing with a rare disease. It's one of the reasons why, though I'm optimistic about the electronic health record, I do wonder, because electronic health record has what the doctor wrote down that the patient has, not what the patient said the patient had. And doctors, not inappropriately, are trained to write down things which affect treatment or care. And so if the patient says something which, I have this symptom or that symptom, but it's not judged pertinent to the care that they need to get, they need to deliver in the hospital, wherever, they're not going to write it down. So that's why natural history is so darned important. But if you ask, when I try to motivate people, investigators, to do this, I tell them, do you know what the FDA is going to ask you? If you go to them with an intervention, what does the FDA ask? They're not going to ask, so I'm a neurologist, right? Neurologists love to measure things that have no relevance to the patient whatsoever. But we can measure them. And so we love doing that kind of stuff. We're famous for it, actually. But if we go to FDA with that, they're going to ask, well, does the patient feel, function, or survive better because of this thing that you measured? And if you can't say yes, they're going to reject your drug. So feels, functions, and survives. Now, of those 3, think about it. The only one that any doctor can unequivocally diagnose is survives, right? Most doctors are able to tell whether a patient is alive or not. But how a patient feels, can't do that. How a patient functions, can't do that. So those are patient-reported outcomes. So if you don't care about anything else, if you want to get your drug approved, you got to listen to the patient and document it. But most people, I find, don't know that.
Right, the patient impact, that's what matters, and that's what the regulators want to know too.
Yes, of course, but it's not what gets you a paper in a prestigious journal, which gets you tenure. So I'm not.
It does.
So it's just the system that we have, and so realize that's the system.
Right. Well, we have lots more questions, but on that note, we could go for hours probably.
Yes,
Chris,
I know, and I'm sure we will all enjoy it.
Just
We're all
fantastic.
equally passionate about this space.
Oh, the comments that are coming through, just best keynote ever, just fantastic content. People are so grateful, Chris. Thank you.
Thank you. It's been great to be with you.
For those who continue to ask questions, we'll see if we can get those answered. So we have a couple more housekeeping notes before we completely close out for the lunch break. We have poster presentations, okay? So if you would like to chat with our poster presenters, please go to the list of chat rooms on the left of your screen and search for either their name or their poster title to enter their chat room. And to view the posters in the poster booth, you'll click on Documents and then you'll click View. I also want to remind you how to get back into the agenda when you return. Okay, so you need to click back onto the auditorium in the lobby. You'll click on the event logo. Agenda will pop up and you'll need to click the orange Join button for the session you want to attend. So I'm just going to say that real quickly again. You'll go into the agenda, you'll click back into the auditorium, you'll click on the event logo, the agenda will pop up and you'll need to click the orange button that says Join for the session you want to attend. We're going to put a post about that in the chat as well. So thank you again to Chris. Up next from 12:30 to 1:30 Eastern time, please enjoy an hour break. And make sure you visit the poster hall and networking lounge, and you'll return back to the auditorium at 1:30 Eastern time for the next session. So please enjoy, and thank you again to all of you.
Thank you.